Hi everyone, today I'll be covering how to do single-cell analysis with ScanPy, an alternative to the single-cell analysis with Seurat and R. So step 1 is to install ScanPy via the terminal. You can use pip to install ScanPy with one line. Installation should be fairly quick if you have some of the other modules already installed, such as Pandas and NumPy. Next, you can open up a Jupyter Notebook page to start your analysis in. The Jupyter Notebook will open up in your browser, and you can simply switch to that window, start a new Python 3 notebook since ScanPy is only compatible with Python 3, and give it an informative name. Step 2 is to download example data. You can download a dataset from 10x Genomics. They have a lot of single-cell expression datasets available. You just enter your email, name, and institution, and can download the data. Here we'll be looking at a dataset of 3,000 PBMCs. We'll download the filter gene cell matrix and start analyzing with ScanPy. So now I'll walk you through a notebook that has a brief analysis of the dataset. This is largely taken from a tutorial available through the Thais Lab, which created ScanPy. First, we'll import the dataset, and you should be able to do that if you already have the downloaded dataset of the 3K PBMCs. Just unzip it in your download folder. Next, we'll be pre-processing the data. So we'll look at the number of mitochondrial genes, and we can visualize the number of genes, counts, and mitochondrial genes in each cell through the simple violin plot. Next, we'll remove the cells with too many genes or too large of a percent mitochondrial genes. So we chose 2500 and 0.05 as our cutoff, similar to Seurat. Next, we'll be assessing the highly variable or highly differentially expressed genes in the dataset. So we can do this with a few simple commands available from ScanPy, and we find that about 13.4% of genes are highly variable. So now we can filter out the non-highly variable genes in the dataset and retain only 1838 genes. Filtering out the non-highly variable genes will allow us to do our analysis only on the likely most meaningful genes. So next we can just logarithmize the dataset by taking and then we'll regress out the effects of the total counts per cell for genes and the percent mitochondrial genes expressed in order to better analyze the different cells and reduce the potential batch effects of these two variables. After this filtering has been done, we can do some data exploration using PCA. So here we're visualizing the first two PCs colored by a gene CST3. And we see a few distinct clusters. We can also look at the variants assigned to each PC, and we see that the first 4 or so PCs seem to be the most significant. Now we'll write this to a results file. We can also do t-SNE, which is a more advanced nonlinear dimensionality reduction algorithm, but which takes a significantly longer time to run than does PCA. Looking at t-SNE, there are a similar number of clusters to PCA, and we can run t-SNE on both the filtered data and the raw data. Next, we'll look at UMAP. This is a much more recent dimensionality reduction technique that's been a lot of interest lately. So it's competitive with t-SNE for the visualization quality, so it's nonlinear, unlike PCA. But it has superior runtime performance. As you can see, I did not speed the video up by 20x this time. In fact, I just slowed it down a little bit to be able to visualize everything. And again, there are similar clusters as the t-SNE and PCA showed for the genes. That I chose to show here, the 3 genes recommended by Seurat and ScanPy. Okay, and next we'll be doing some clustering, but first we have to install a couple modules on the terminal. So here first we'll install iGraph, which is required to do the clustering. And this does take a little bit of time, so you have to be patient. And then the second module that we'll install is called Louvain. So this is because the clustering type that's performed is called Louvain clustering, and you can read more about that in the original paper, which I'll link in the next screen. So the reason that we want to do clustering, even though clusters are able to be properly visualized on t-SNE and PCA is just to have a more rigorous mathematical way of defining what a cluster is, not only just based on visual parameters. So as you can see here, I did Louvain clustering, and this automatically clustered the cells on the t-SNE projection. In addition to t-SNE, we can visualize the clustering results on UMAP, which comfortingly look very similar to t-SNE, even though UMAP took significantly less time, as you saw from the video. Next, we'll write the clustering results to our file. We can also look for marker genes in each cluster, which can help identify which known cell types the clusters correspond to. And you can take a closer look at each cluster and see which genes are ranked for each cluster as most important, and the relative effect size compared to the other genes in each cluster. We can also do this ranking with a different method via logistic regression, and you can compare the two in a more detailed fashion if you would like to. Finally, we can visualize the top genes per cluster using a simple data frame and look at the scores for those as well. You can also use a violin plot to look at the more detailed view for a single group. Here I chose 0 versus rest. And finally, you can compare a few genes of interest across all groups. Okay, so now that we've shown you how to use ScanPy to do some basic single-cell analysis in Python similar to Seurat and R, we'll be using diffusion pseudotime that was developed by the Thais lab and is available only in ScanPy and not in Seurat to order a dataset, a qPCR dataset of early hematopoietic development. This section of the tutorial was also largely adapted from a tutorial available from the Thais lab. And if you haven't heard much about diffusion pseudotime before, I really encourage you to look into it. There are a lot of alternative competing approaches available from other labs as well that have come out in 2017 and 2018, and I'm sure will continue to come out because ordering cells along developmental trajectories is an outstanding problem in the field and is certainly of interest since currently single-cell RNA sequencing methods are destructive and it's not possible to experimentally view a single cell as it's developing, but rather it's of interest to computationally order these trajectories from populations of cells when the populations are at different stages. So this approach does, however, require some prior knowledge. As you saw, I just defined a root cell in order to do the diffusion pseudotime. We can compute neighborhood relations among single cells quickly. And here you see the trajectories. So they're automatically identified diffusion pseudotime groups, and you see diffusion component 1 and 2 projected there, and they're colored by the experimental groups, so it seems to show fair agreement. We can also view in a more detailed fashion expression of all the genes along the diffusion pseudotime trajectory. And finally, we can annotate the cell groups according to known cell states, and then save the results. Here it was already known that these cells were either undecided endothelial, erythrocytes, trunk, or endothelial cells. Well, I hope you enjoyed this tutorial, and thank you for following along.